Hello everyone, this is Rohit Shankar and today we are going to see molecular docking. Now, molecular docking and overview. Now, first we are going to understand the basics of molecular docking. What is docking? Approaches for docking. Types of docking. Autodock. Now, Autodock it is an software where we can perform molecular docking. So we are going to understand Autodock. What are the requirements for Autodock? How to perform molecular docking in Autodock? Conclusion and references. Now let's see basics of docking. Now receptor is also called as host. It is also called as lock. It is a large molecule and it receives another molecule. Ligand it is a small molecule and it binds with the receptor. Docking it is a computational simulation of a candidate ligand binding to a receptor. Binding mode it is a study of how ligand is going to bind to the receptor. Pose. Now pose in which mode of receptor binding with ligand that we are going to study. Scoring. It is an analytical process where we have to determine the interactions of ligand to receptor. Ranking. It is an analytical process where we have to determine the target of ligands and how ligand is going to bind with the target. And docking assessment where we have to interpret our result, that is interpretation of our results. Now, what is docking? Docking it is an analytical method which predicts the preferred orientation of one molecule to a second molecule when bound to each other and to form stable complex. Knowledge of the preferred orientation in turn may be used to predict the strength of association. For example, scoring functions. Schematic illustration of docking a small molecule ligand to a protein target producing a stable complex. Example, docking of a small molecule which is shown in green into a crystal structure of beta 2 androgenic G protein which is shown in black coupled and it forms PDB3SN6. Now by this figure we can easily understand the docking. So docking is nothing but the when ligand binds with the target then they forms complex. That complex is nothing but the docking. So for that purpose we have to understand the ligand and target structure. Let's see. The associations between biologically relevant molecules such as proteins, peptides, mononucleic acids, carbohydrates, and lipids play a central role in signal transduction. Furthermore, the relative orientation of two interacting partners may affect the type of signal produced. Therefore, docking is useful for predicting both the strength and type of signal produced. See this figure. Now, with the help of this figure, we can easily understand that we can predict the molecule interactions in the molecular docking. Now, here the how the ligand is going to bind with the receptor and what will the interaction in between them, that we can easily understand. Now, approaches. There are mainly two approaches. One approach uses a matching technique that describes the protein and ligand as complementary surfaces, and another one is it approaches the simultaneous the actual docking process in which the ligand protein pairwise interaction energies are calculated. Now, first one simulation approach. Now here the target and the ligand are separated by physical distance at first. Then when their activation energy activates themselves, then they will come together and they will form the complex. That energy is known as a total energy. Next is shape complementarity approach. Now here ligand and the target has their specificity, that is space shape specificity. According to that, they will bind with each other and they will form complex. Now, types of docking. Docking tools employ search algorithms such as genetic algorithm, fragment-based algorithms, Monte Carlo algorithms, and molecular dynamics algorithm. Besides this, there are dock, gold, flexes, and ICM. There are various kinds of molecular docking procedures involving either ligand or target flexible or target based upon objectives. For example, in many cases, ligand will be flexible and target will be rigid, or vice versa. Ligand will be rigid and target will be flexible, or in some cases, both will be flexible or both will be rigid. How to perform molecular docking? The docking process involves two basic steps. First, we have to understand the position, orientation, and the binding affinity of the ligand with the proteins and the structure, orientation of a proteins. Usually, the structure has been determined using different biophysical techniques such as X-ray crystallography, NMR spectroscopy, and cryo electron microscopy. The protein structure, any database of potential ligands serve as a inputs for docking program. The success of a docking program depends on two components. First one, search algorithm and score function. And these 2 points are depends on the just only ligands and the proteins. Now this example, protein TGF type 1 ligand citrulline minimum binding energy is minus 7.5. Amino acids are available. Now Autodock. Now Autodock, it is an basic bioinformatic instrument or software where we can use for the interactions of molecular docking. Autodock has been developed to provide a procedure for predicting the interactions of small molecules with the macromolecular targets, where we can separate different compounds and we can identify the new compounds. How to use Autodock? It is an excellent non-commercial docking program. Stochastic Lamarck-King genetic algorithm for computing ligand conformation. There are different steps. First step is to retrieve required ligand and target protein files. Then this file convert into PDBQT format files. Then perform molecular docking using SizeWin, and finally we have to analyze the results. Requirements: Windows XP or Windows 7, MGL tools, SizeWin, Discovery Studio Visualizer, binary files, Java. These 6 files are required. How to perform? First, we have to retrieve the ligand and protein data files. Okay. Then we have to prepare PDBQT format files of ligand and proteins. Then using SizeWin for molecular docking, we can perform. Then analyze your retrieving ligand enzyme interaction results. This will be the results. Conclusion. Now here Autodock is a non-commercial, very easy software where we can use for the molecular docking. And the present presentation reveal different aspects of the molecular docking. For example, basic techniques, how to perform molecular docking, what is Autodock, and how to use Autodock. These are the references. If you have any question, you can comment or you can mail me. Thank you.